When we work with one categorical variable, we perform tests that fit observed data to expected data. When we work with two or more categorical variables, we essentially do the same thing. We're going to compare observed to expected data, but usually our expected is going to be derived from the data itself. Oftentimes, with these two categorical variables, what we are most interested in is the association between those two variables. So if we look at one variable and we are in one category, does it make it more likely that we are in a specific category or group for that second variable? This test of association is also called contingency analysis. So with these contingency analysis, we are typically working with two or more categorical variables. For example, let's say we have age and sex. And with age, we're working with age categories. So let's say young, juvenile, and adults, and then sex, we're working with male or female. What we want to know is the ratio of sexes equal across the different ages? This is our test of association, and it's equivalent to a test of independence. Types of questions that we could ask and answer with a contingency analysis is this. Is classification for variable 1 independent of the classification for variable 2? Also, are the classifications for variable 1 and 2 independent of each other? Or are the classifications for variable 1 and 2 associated with each other? We could also rephrase our questions to place less emphasis on independence and more on how variables vary. So for example, does sex ratio vary by our species, or does it vary by age, etc.? The calculations that we can use are the exact same as what we use for goodness of fit tests. So if we do a chi-square contingency analysis, we utilize the same equations as a chi-square test. If we're doing a g-test contingency analysis, then we use the exact same equations as a g goodness of fit test. The expected values are going to be derived from probability theory. So our assumption, our null hypothesis, is that our variables are independent of each other. Are we going to calculate these by hand? Nope. R will do it for us. So before we get to contingency analysis, I want to take a detour and talk about margins. So margins are simply the row and column totals. So if we have a table, a frequency table, we would have number of rows for one variable and then a number of columns for the second variable. Our data are in that table itself, but then we could have row totals. So we add up all of the columns across a row, and then we also have column totals. Those are the margins. If we want to view those totals, we use it by stating add margins to our table. And we can generate this table by giving any number of variables that we're interested in. Now with these margins, we can see that they are row sums and column sums. And when we run our analysis, we can fix one, both, or none of these totals. And this leads to different model types of contingency analysis. I'll state right off that the rest of this is FYI, dealing with these different model types. So the rest of this slide and all of the next slide is just FYI. So what is a Model 1 contingency analysis? Well, these contingency analyses do not fix any of the margins. So they only fix the total number of samples in our table. So our grand total is what I would say. So let's say we sampled 100 individuals. We're going to fix 100 individuals. We don't know what the underlying distribution is in each category, and we don't really care. We will test assuming that we will always have 100. A Model 2 is going to fix one of our margins. So it's either going to fix all of our row totals or it's going to fix all of our column totals. So for example, let's say we sampled 100 individuals of each of two species. What we could say is with our null hypothesis, we would fix that 100 individuals for each species and then let the different categories for variable 2 vary. For a Model 3 contingency analysis, both of our margins and the columns are fixed. So if we sampled 100 individuals of each species for the first variable, and then we measured second variable as whether or not individuals live or died, and we stopped our experiment once we had 50 individuals, then our null hypothesis assumes that we will have a total of 100 individuals for each species and 50 individuals dying for that second variable. Why does it matter? Well, each of our tests make a different assumption. So Fisher's exact test, which we'll use when we have exactly two groups and two variables. So two variables and two groups within each variable. That's a Model 3. Both margins are fixed. And that's different than if we do a Model 1 or a Model 2. So G or chi-square tests are pretty much always either a Model 1 or a Model 2 test. Randomization tests are typically a Model 2 test because we're going to shuffle one variable and keep the other variable constant. What about randomization for Model 1? Well, those are kind of difficult because we don't technically know our total sample size. So it's kind of hard to conceptualize randomization. Now, does this even matter? No, probably not. I think the only case where it matters is with our Fisher's exact test. Because a Fisher's exact test, since it is a Model 3, tends to be a little bit too conservative than a Model 1 or a Model 2. So if we did a Fisher's exact test and saw, let's say, a p-value of 0.08, well, is that really because they're not associated, or is it because Fisher's exact is too conservative and both the G or chi-square would find a significant difference? Regardless of Model 1 or Model 2, if we do a G and a chi-square test, we're going to get basically the same answers. The only caution would be with randomization tests. We're not going to get into any of this. We're not going to get into it for this class. We're not going to get into it with any other statistics course, and really, even in research analysis, we don't really care, not for at least a lot of our biological data. But I just present it for some completeness. So let's talk about what happens when we collect our data. So we get our data, we have two categorical variables. First thing we're going to have to do is illustrate our data in some way. We've got three different ways to do it. So the first way is frequency table. We can do that. And our frequency tables are pretty nice because we can give it both of our variables. So the table function can take variable 1 and it could take variable 2, and it'll output a table. If we have three variables, it'll output a series of tables. Very easy to do. We can also make a frequency table by referencing the column numbers in our data table if we have a very large data table. Normally we can just say data $variable, data $variable, and it'll work. So frequency table is just that, it's a frequency table. Our plots, or our graphics, could be either a mosaic plot or a grouped bar plot. Now a grouped bar plot is one that you've probably seen before. We can do this in R by passing a frequency table to the bar plot function, and it works okay. Excel makes even better bar plots, and these are the ones where we have a group of bars touching each other, and then a space, and then another group of bars touching each other, and then a space, and so forth. I'm sure you've seen this. Nice thing about Excel is that we can modify the bar colors easily. We can do colors, we can do cross-hatching and all that other stuff. And we can also add error bars. So we can do bootstrap confidence intervals, just like we would do for a one categorical variable, and then add those in Excel. The plot that I like to make in R is a mosaic plot. Now the information in a mosaic plot is displayed as the area within our bars. And our bars are going to be scaled to 1. So it doesn't matter if we have 1,000 individuals for one variable and only 100 for another. They're going to scale it accordingly. They're going to scale everything to 1. Now the mosaic plot's really good at showing independence or lack thereof between two variables. Because if we have independence, then our bars are going to basically line up, and the breaks in the bars are going to line up to show us that the pattern that we see in one group is the same across all groups. The mosaic plot separates the bars too, so it emphasizes qualitative nature. Now, how we make mosaic plot is to use the mosaic function in the VCD package. We can use mosaic plot that's in the base package, which works. However, I really like this mosaic function in VCD because it adds a couple more details to the plot. So how we do that is use it as a function. And with our categorical variables, we don't really have independent and dependent variables. Rather, all of our categorical variables are these independent variables. So we would use our tilde sign or squiggly, and then variable 1 plus variable 2, specify the data frame where you would find those variables, and then do shade equals t and legend equals t. These will shade the bars based on our Pearson's residual, and then it would also add a legend. So we'll see this. So here's an example. In our ecology class, we went and looked at the distribution of ragweed in a grassy area. While doing this, we also recorded presence or absence of dandelions. So we can make this table where we have a grid where ragweed are absent and dandelions are absent. Some dandelions are present. And then we have grids where the ragweed was present, and again, some dandelions were absent, some were present. And we get a total of 180 grids that were counted. We want to know if ragweed and dandelions exhibit an association, which means if ragweed are absent, does it make it more likely that dandelions are present or absent? If ragweed are present, does it make it more likely that dandelions are absent or present? Now with this association, we could have a positive association where our absent, and present, are more likely to occur. Or we can have a negative association where the presence of one makes it less likely we find the other, and so forth. So these two would be more than what we expect. So with our question of are these associated, we want to proceed by doing a chi-square and a g-test. Now we can do these by hand. We can take these observed values and then we can calculate expected using probability theory and do a regular chi-square and g-test. So once we do that, we have to adjust our degrees of freedom. And our degrees of freedom for all contingency analysis is the number of rows that we have -1 times the number of columns that we have -1. So in this example, we have two rows. So 2 minus 1 is 1. So this one would be 1. And then we have 2 columns. So 2 minus 1 equals 1. So 1 times 1 gives us 1 degree of freedom. So let's go through how we would actually do this in R. Well, in order to do a contingency analysis in R, we have to first convert our data table into a matrix. And a matrix is simply an array. It's an array of data arranged kind of like a table, where we have rows and we have columns. So in order to get our data into a matrix, what we're going to do is issue the matrix command. And the matrix command is going to take a vector of numbers and create a matrix. Now, how does it know how many rows and how many columns to create? Well, we're going to specify either the number of rows that we want or the number of columns that we want. And then we'll specify if we entered our numbers by row or if we entered them by column. So for example, in this table, we could enter our vector like this: 25, 35, 45, and 75 in order. 25, 35, 45, 75. So there's our vector. How many rows do we have? Well, we've got 2 rows. So I would do n row equals 2. And then we loaded this vector by row. By row, 25 and 35. That's our first row. And then 45 to 75. So we're telling R to take this vector of data, make it a matrix that's going to have 2 rows, and these data appear by row. So when we do this, we're going to save this as OBS, observed, and then once we run that command and issue the OBS function, you see that we get this matrix of data, and it appears exactly how it appears here. So go ahead, pause the data, get these into our studio. So here's my code. I ran it. I checked my matrix. That looks exactly how it appears. And now I want to run a chi-square contingency test. Now, if you go back to the previous PowerPoint, you would see that we had a couple different ways to write this chi-square. One way is to give it a matrix. So chi-square.test, OBS, correct equals false. I run it, and it runs a Pearson's chi-square test. Here's a test statistic. Degrees of freedom is 1. P-value is that. Is the degree of freedom correct? Yep, it is. We had 2 rows and 2 columns, which makes our degrees of freedom 1. So based on this, is our ragweed and dandelion independent? Yes, they are. Ragweed and dandelions are not associated. And we ran a chi-square test. That's what we run, chi-square contingency analysis. Our chi-square test statistic is 0.29. Degrees of freedom is 1, and our p-value is 0.59. What's the expected values? Well, for lists, we can view what our expected values are by issuing the name command. When we see the names, you can see that we have all of these things that we can reference using the $sign command. So I can do chi-square.test, OBS, correct equals false, and do $sign and specify expected. And when I run this, this gives me my expected values. These are the values that we would then, that the chi-square test uses to generate our chi-square value. So I have an example write-up, just like what I did in the R Markdown. So you can see it's not very difficult. If we have our data as a table or as a matrix, chi-square test will do it correctly. What about g-test? Well, in order to do a g-test, we'll use the VCD package. And in the VCD package, we have a function called associatstats, or association stats. It's a test of association. It's going to do the exact same thing as a chi-square test, similar idea, all right? Meaning we don't have to calculate our expected values by hand, we'll let R do it. Now the nice thing about the association stats function, or associatstats function, is that we only have to pass it a matrix of observations. So let's do that. In R, make sure you have the VCD package loaded. So I had library VCD up in my libraries. It's loaded. And then let's run our g-test. So it's going to be associatstats and OBS, and it gets this. Now, like our good fit function previous, it's going to report 2 lines. It's going to report a likelihood ratio line, and it's going to report a Pearson's line. This Pearson's line is the chi-square test, chi-square contingency analysis. That chi-square, 0.29221, is exactly the same chi-square that we obtained from chi-square.test. This likelihood ratio line is the g-test. So, if I asked you to run a chi-square and a g-test on a contingency table, you can run this associatstats and obtain both of them very, very easily. Notice degrees of freedom is 1. That is exactly what we were expecting. What are these phi coefficients, contingency coefficient, Kramer's V, and so forth? Well, these are associations. This is kind of like a correlation. The more associated we are, the higher the value these are. We're not going to mess with it, but I just wanted to explain why you see them. So just like before, based on our test statistic, our p-value, we would conclude that ragweed and dandelions are not associated, or that their presence is independent of the other. What's the Fisher's exact test? Well, the exact test is just the same. It's testing association. Is one variable associated with the other? However, it's going to be an exact test. It's an exact test because it calculates an exact probability, and it does so using a hypergeometric distribution. This hypergeometric distribution is basically calculating how many different combinations of tables, contingency tables, there are given a data set, or given the number of objects in our data set. So for example, going back to this matrix, this represents a contingency table. What Fisher's exact test will do is utilize the hypergeometric function, hypergeometric probability, to figure out how likely it is to get this combination, this exact table, and how many more extreme table, and the number of extreme tables that we have. So recall the definition of a p-value. The probability of getting data as extreme or more extreme than what we have actually observed just by chance alone. Well, the chance alone for Fisher's exact test is that hypergeometric distribution. Do we need to know that hypergeometric distribution? No, we don't. Are there assumptions to this test? No, there's not. It's an exact test. We will calculate it. Now, Fisher's exact test is really only practical when we have a 2x2 table. So 2 categorical variables, and each variable only has 2 groups. How do we do this? Well, we use Fisher.test, where our X is the matrix of observations. So we can use it in R using Fisher.test OBS, and we get this output. I'll note that with Fisher's test, we do fix the row and the column totals, so those don't change. And that's kind of unlikely for real data, but that doesn't seem to matter. The general scientific community is okay if you run a Fisher's exact test to see if 2 categorical variables are associated. So using the same test, Fisher's exact test generates a p-value of 0.6284. So again, ragweed and dandelions are not associated. There's no test statistic associated with this. There's no degrees of freedom associated with this. This is an exact test. So let's do a practice. You trapped skunks and recorded the sex of each. And this is our table. So our question, did we see the same sex ratio for both skunks? So did we see the same sex ratio for both? Let's change that. Let's put that to 60. All right, so here's our table. What this is asking is male-female independent of our species of skunk? So first thing we're going to have to do is make a matrix. That's going to be our first step. So how do we make a matrix? Well, I'm going to use the matrix function. And what I'm going to do is read it across the rows, so 10, 20, and then 25, 60. Number of rows that we have is 2. By row, yep, it's true. And then I'm going to view it just to make sure that I've entered it in correctly. That looks just like what we did. All right, so now what we want to run is the contingency analysis. So we have a couple different ways to do this. So the first is the chi-square test, which would be chi-square.test and OBS incorrect equals false. We could also run our g-test using associatstats, which gives us that, or we can run our Fisher exact test, which is Fisher test OBS. All right, so chi-square test generates a chi-square of 0.16, 1 degree of freedom, p-value 0.66882. That's the same that we get here. G-test is very, very similar. And the Fisher's exact gives us a little bit higher p-value. That's our conservative nature. So based on all of this, no matter which one we choose, we conclude the same thing. Did we see the same sex ratio for both skunks? Yes, we did. Sex is independent of species. So that's two-way contingency analysis. So each group has two categories. But if we have more than two categories in one or both of the variables, the tests are run exactly the same way. Degrees of freedom is the same way, number of rows minus 1 times the number of columns minus 1. We can still use chi-square.test, we can still use association stats, and so forth. So everything's basically the same. So for our example, let's utilize seatbelt.csv. In this data set, it asks a question, is severity of injury independent of seatbelt usage? So what I want you to do is download this file, import it into R, create a summary table, and then answer this question. So going back to my R, I read in my seatbelt data, and I called it seatbelt. It's here, and I looked at the variables. Now, while we can pass a data frame to seatbelt, or to the table function, I don't recommend it. Because if you have more variables than the two that we're going to analyze, it's going to really produce some weird tables. So my recommendation is always to pass the variables that you want to include in our contingency table. So here's mine. I did table seatbelt worn and seatbelt injury. So the first variable is our rows, the second variable is our column. And what we see is that we've got none, minimal, minor, and major all out of order. So one thing that we may want to do is reorder our injury. Do you remember how we did that? Well, we're going to reorder injury and we're going to save it back into the same variable. So we do that using the factor command. We're going to factor seatbelt injury, where our levels is equal to how we specify them. So I'm going to say I want none, minimal, minor, and major. I'm going to run that, and then when I run it, now I get it in order. And it gives us a better way to actually visualize it. So our question, is injury associated with seatbelt usage? What we're looking for is, do we see many more injuries when we don't wear a seatbelt than when we do? And just looking at the table, it's kind of hard to say because we have a different number of row totals and sum totals. So this is where contingency analysis really shines, because it will calculate our expected for us. So, does it matter which one we use, chi-square or g-test? Nope, not really. So, what we can do is use associates to generate our chi-square. So, we can use our chi-square, 59.22, or we can use our g, 62.83. And in both cases, you should see that we have a small p-value. Now, is our degrees of freedom correct? Yep, it is. So number of rows is 2, so 2 minus 1, times the number of columns minus 1, so 4 minus 1 is 3. So 3 degrees of freedom. So in both cases, no matter which one we use, injury does differ based on seatbelt usage. So if it looks like if we don't wear a seatbelt, we are more likely to suffer major and minimal injury than if we do wear a seatbelt. How do we look at our expected values? So we said chi-square, we can use chi-square $expected. And let's look at those. So we did association stats. Let's run our chi-square test. So we did chi-square.test. We're going to pass it table, seatbelt $worn, and seatbelt $injury. And we want correct equals false. I need to spell it correctly. All right. So what I can do is look at expected. And we get these values. So we expected 289.88 major injuries, but we observed 303. We expected 55 major injuries when we do wear a seatbelt, but we only observed 42. So it looks like these things are switched. We saw less major injuries when we wore a seatbelt, and we saw more major injuries when we did not wear a seatbelt. See somewhat of the same thing for minor. We saw more than what we expected when we don't wear a seatbelt, and we saw less than what we expected when we do wear a seatbelt. Now that's for a chi-square. Does VCD have a similar function? Yes, it does. That function is independence table. So this generates a table of independence, which produces the exact same values as we had in the chi-square. So in the presentation, I reordered them in reverse, but we get the exact same values. So in R, we would use independence table and pass it our actual table function to get our expected values. So here's this one after we've reordered, and you can see they are exactly the same number. So just visually, we can go one by one and make the comparison between our observed and expected and come up with these conclusions. When we don't wear a seatbelt, we get more injury than what we expect. And we have fewer none than what we actually would expect. And when we do wear a seatbelt, we get more, in a case where we have no injuries, but less injuries than what we expect. Now this is all fine and dandy, but it doesn't really tell us which ones are driving our difference. For that, we really need to see the relative loadings. And this is where the Mosaic plots really come into play. So we can visually display the data using Mosaic plot function or the Mosaic function. The Mosaic function takes on this type of format where we put the squiggly and then both of our categorical variables. We use shade equals t so that it adds shading to show the largest deviations from our expected. And what it's going to plot will be the Pearson's residual. So that same Pearson's residual that we observed from the good fit table in the last presentations. What this Pearson's residual displays is a relative contribution to the chi-square statistic. So the larger this residual, the more it contributes to this chi-square. So for this injury, or for our seatbelt data set, I would generate the plot just like this. Just like this. Mosaic, worn plus injury, data is seatbelt, so these two variables are found in the data set seatbelt. Shade is T and legends is T. Then we get this. Anything in the gray means we have a small Pearson's residual, so it has very minimal effect on our overall chi-square. In essence, our observed and expected are pretty close. So no seatbelt, no injury, it's kind of what we expected. No seatbelt, minimal injury, it's kind of what we expected. All right. If we have positives, then and we have what we mean is our residuals on the positive side of things. So we're seeing more than what we expected. So when we wear a seatbelt, we have more nones than what we actually expected. When we don't wear a seatbelt, we had more of these, this category. And I can't see it because it's all just kind of jumbled together. I think that's minor, more of the minor than what we expected. If we're into the pink and the red, that means we're seeing less than what we expected. So when we wear a seatbelt, we see very much less of these more major injury categories than what we actually expect. So the blue means we're seeing more than what we expect. The red is we're seeing less than what we expect. What's important are the darker shades. So there is one more blue, like a dark blue, that we could have seen. Anytime we have a dark blue or a dark red, that's a strong indication that that category is what's driving our difference. These, the lighter shades, are ones that suggest that it may be significant. So let's go ahead and do this mosaic plot in R. So that was on our presentation. So let's do our mosaic plot here in our markdown. So it's called Mosaic. We're going to use the tilde sign. We're going to use worn plus seatbelt. Worn plus injury. Our data set is seatbelt. Shade equals T. Legend equals T. And we get this. So just to see if I can get this bigger. No, I can't. So we get basically the same thing. Shows us which ones look different. Similar mosaic plot can be obtained by using the mosaic plot function. So mosaic plot, that's in our base package. It can take the same type of stuff, but legend is by default. That's why we got that error. So when we do this, it shows the same type of stuff. I just think the mosaic function looks a lot better than the mosaic plot function, even though the graphs basically illustrate the same thing. So we found a difference. We looked at the plots. It looks like we've identified the different groups. Are there post hoc tests that we can run? Oh yeah. So think back to what we did with ANOVA. ANOVA was our omnibus test. We're testing to see if the group means differed. If we get a significant difference, then it tells us that the means are not equal, but it doesn't tell us which means differ. Perhaps it's only one mean that differs from the rest. Perhaps two means differ from the rest. Perhaps all means differ from the rest. What we had to do after that ANOVA was to do a post hoc test. Tukey's HSD or some other multiple comparison test that will tell us which means differ. Goodness fit tests are the same way. After we get significance, it tells us that our observed differs from what we expect, but it doesn't really tell us which ones differ. So what we can do is do a series of post hoc tests that analyzes individual labels and or groups within our data. When we run this, we have to realize that anytime we do these repeated tests on our data, we're going to increase our type 1 error. So with each test that we run, we're going to have to adjust our p-values accordingly. We can adjust our p-values using the same thing that we did when we ran our pairwise t-test and pairwise permutational ANOVAs and so forth. We can use Bonferroni, which is simple but conservative, or we can use Holm, which is my preferred method, and it's a little bit more powerful than Bonferroni. What's important, though, is that these adjustments are based on how many comparisons we make. If we have a lot of different comparisons, it's going to make it harder for us to actually detect where the differences are. So when we when we do these post hoc tests, what we really try to do is minimize how many comparisons we're going to make. I have Seaboard. I used to spend some time talking about choosing how many tests we want to run, but we're not going to do these as post-hoc tests. We just learn how to do contingency tests. So if you do get in the situation where you do this for your research or for some other project in another class, please come talk to me. I'll help you out with these post-hoc tests. So here's some practice. A particular antelope species was examined for infection by GeoHelminths. The age of the infection and infection status was recorded, and the total number of parasite species in the gut was recorded. These data are in antelope.csv dataset. So when you download this dataset, you will see that we have 3 variables. We've got age, which refers to calf, juvenile, non-reproductive adult, and reproductive adult. And then we have status, which is uninfected or infected. And then we have a third column, which is species richness. So our question, is infection status independent of our age? So what I want you to do is generate a display, either do a bar plot, mosaic plot, or table, and then answer the question using both a chi-square test and a g-test. Alright, so let's see what you've done. So our preference would either be a frequency table or a mosaic plot. Frequency table's fine. It allows us to see how many individuals we actually have. So you can see that we only have 2 infected calves and 17 uninfected. So you can kind of looks like we're going to have some small samples, which may influence our decision of choosing a chi-square or a g-test. This is the mosaic plot, which I did infection plus age and produces this plot. Again, it's not that great. I prefer mosaic plot, so in RStudio I've got my table. Here it is. Age and infection. And then I do this, and I think this looks a little bit better. Now, these things are smushed together. We could correct that, especially if we do it outside of the R Markdown window. So I can run it here. And then as I increase this size, it starts to spread things out. And we can modify it, again, reduce the margins and everything. And, if you need to do this, come talk to me. I'll help you get these images transferred over. If we didn't like how it appears, so if we wanted infection and age spread out across, all we have to do is reverse the order. So INF plus age. Yep. Do that, and you'll see that it puts it sideways. Alright, so we look at this, and just right now it looks like I may be seeing more uninfected than what we expect for the calves, and more infected than what we expect for our last group, which is this adult. Now, importantly, if we compared our mosaic plots, let's go back a few. In this mosaic plot, we saw there was a significant association. And that significant association is depicted by having these bars shifted and not lining up. The same thing is here. Look at this calf 0 group. It's really big here, but really small here. If we were randomly associated, then all of our bars should kind of line up in the categories. So we should have this bar have a corresponding bar right next to it, and this bar have a corresponding bar next to it. So right now, just looking at the mosaic plot, I fully expect to see an association. So our step 2 is to run our tests. I think my preference would be a g-test. And I say it's a g-test because I'm expecting some smaller counts. And our assumption for these contingency for the chi-square and the g-test apply, even though we're doing a contingency analysis. So chi-squares don't do well with small observed and expected values. Well, these are going to be fairly small observed and expected values, so I would lean more towards the g-test. So I look at the g-test, likelihood ratio, that's my g, that's a p-value. So does it look like infection and age are independent? No, it looks like infection status does vary with age. And then we ask the question, well, which age groups differ from the expected? Our options are to run a post hoc test, which we won't do, or we can look at the Mosaic plot. So here's our Mosaic plot again. What it looks like after we would state our interpretation that infection status varies with age, and then we would look at this plot and say, yes, calves are less likely to be infected, or they're less likely to be infected, or more likely to be uninfected than what we expect. We saw many fewer infected calves than what we expected, and we saw many more infected adults, reproductive adults, than what we expect. So it looks like when you're a reproductive adult, you're more likely to be infected. When you're a calf, you're more likely to be uninfected. And that would basically be our interpretation and our write-up. So our write-up would include the g-test, our g-test statistic, 3 degrees of freedom, and the p-value, and then the post hoc would just be an interpretation of our Mosaic plot. Do we have randomization tests? Yeah, we can run a randomization tests anytime we think our assumptions may have been violated. The most likely violations will be when we have a low observed or low expected counts. When we have those, then the chi-square test statistic is going to deviate from our actual chi-square distribution. G-test will also deviate, but not as severely. So oftentimes you can get away with just running a g-test. But if we have really small values, or if we have a lot of categories that have small values, then even a g-test won't work. So for that, we're going to have to do some sort of randomization tests. Now, how do we do these randomization tests? The chi-square.test function had that simulate.p.value test. And we could use this, but it's more like a permutation test when we work with a contingency table. It just basically shuffles the numbers around in our table itself instead of generating just a completely new random table. In this way, it makes this test much more like a Fisher's exact test, which really isn't very realistic, especially if we're trying to test for association. So a better approach is to just avoid this chi-square.test part and do our own randomization. Our own randomization would involve taking one of the variables and shuffling it to generate our own null distribution. And then what we would do is save that G or that chi-square test statistic and repeat it over and over and over again to get our null distribution. And then we would obtain our p-value by looking at our observed test statistic and comparing it to our null distribution. I don't have a script for this because we normally just do it by hand, and we're not going to do it in this class. So if you ever have a need to do a randomization test, then come talk to me, and I'll help you. I'll help you do it. So this basically wraps up our categorical variable analysis. We worked through one categorical variable and learned how to do goodness of fit tests, and then we added a second categorical variable and did also goodness of fit tests, but in this case we're doing contingency analysis, which is essentially a test of association. In both cases, when we have one categorical variable or two categorical variables, the calculations are exactly the same. The way that we calculate the chi-square and the way we calculate the g are exactly the same. Interpretation is also the same. We obtain our p-value and either our observed matches the expected values or they don't. So there's really not much of a difference. But with both of these, goodness of fit and contingency tests, when we do get a significant difference, then it's really important for us to look back at our data and make declarations about which groups or categories had more observations than what we expect and which ones had less observations than what we expect. We can proceed to do statistical post hoc tests on these, but we won't in this class.